Hello, everyone. So I will be telling you about this latest paper from our lab, where we find large increase in theta rhythm and the emergence of a novel eta rhythm when the rats are running in virtual reality. This work was done by a bright postdoc, Dr. Karen Safarian. The theta rhythm was discovered nearly 70 years ago. If theta rhythm, there are impairments in hippocampal theta rhythm, there is decrease in cognitive performance. If theta rhythm improves, it's related to better performance. It changes neural plasticity, learning, neural representation. So it's a pretty important subject. We wanted to understand what are the mechanisms that can boost theta rhythm. In order to do that, we developed a non-invasive virtual reality many years ago. Just like human beings, perhaps a patient, that's an Alzheimer's patient is being tested with a virtual reality headset. This rat can sit inside a non-invasive virtual reality. He can slip out of that any point. His head is free to move, but he cannot run around in any other place. There's a camera that is placed above, and the rat has to simply go to this virtual pillar to get a reward from a sugared bit of water, and he runs around, and he can now the rat can be tested in a similar virtual reality as human in a very positive way. Now, when we measured the activity of hippocampal neurons and the EEG or local field potential, we measured simultaneously in the real world and virtual reality on the same day. So when the rat's running in the real world, when he's running at low speed or is stationary, you don't get any theta rhythm. When he starts to run on that electrode, you see this very beautiful theta rhythm that was discovered 70 years ago. When he runs in virtual reality, very similar stuff happens. When he's running at low speed, you don't see much. When he runs at high speed, you see very prominent signal. Is this really theta rhythm or something else is going on? So you filter the signal in the 6 to 12 hertz theta band, and you can see most of the fluctuations of the raw field potential in the real world are captured by this theta band signal in green. But in virtual reality, while you see plenty of theta signal, there is something additional that's going on that's not being captured. Once again, when he's stationary, there is very weak theta both in real world and VR. Now, if you filter the signal in an additional band called eta band, which is something we discovered now, which is in the range of 3 to 5 hertz, you don't see much in the real world still, but in VR, that's massive signal. That's this eta rhythm that is captured by combination of theta plus eta. And that's the story. Now, where does this come about? Why haven't we or others seen this before? If you look at the power spectrum from this, you see very clear peak at theta rhythm when the rat is running, not when he's stationary. In the real world, when he runs in virtual reality, you see theta rhythm, but you also see this a novel bump at eta rhythm, so it's real. You can look at it in terms of spectrogram. Here is the running speed in black. Here's the amplitude of theta rhythm. Here's the amplitude of eta rhythm. This is in the real world. You see pretty much strong theta modulation when he's running, but not much eta. In VR, in contrast, you see very reliable increase in eta amplitude when he's running. And that's black trace right there. This is the spectrogram underneath it. You see in real world, pretty much theta rhythm at every place when it runs around. When he stops, you see type 2 theta, which is somewhat lower frequency, but there is no eta rhythm here. Whereas in VR, when he stops, you see you see nothing again. But when he runs, along with theta, you see this eta rhythm at the same time, very reliable. So where is it showing up in hippocampus? It is well known that in hippocampus, theta rhythm shows up in the region in which there are dendrites. In the CA1 cell layer where sharp ripples are weak, you don't see much theta, but as you go away from it, you see more theta rhythm as you go into the dendritic region, both above and below. Eta rhythm, on the other hand, does the opposite. In the cell layer, the eta rhythm is maximum. And as you go away from the cell layer into the dendrite, the eta amplitude reduces. So theta rhythm is in the dendrites in both real world and virtual reality, but eta rhythm appears in the cell layer predominantly in VR. And that's the minority of electrodes which are in cell layer. That's only 50 micrometer thick. That's why people could have missed it. If you looked at the distribution of the amount of amplitude of simultaneously recorded cells versus the depth, there is a very strong correlation. between eta amplitude and self-favorable polarity, which is the exact opposite of theta. So that gives us even more confidence that this is a new phenomenon that's appearing in the cell layer. So who is doing that in the cell layer and why? It turns out that the dependence of eta rhythm and theta rhythm in virtual reality is quite surprising. In eta amplitude shows this dependence in VR. It actually goes down a little bit at low speeds as the speed increases and then goes up. And in the real world, there is no eta, of course. And if we looked at the stats across all the data, these are the two important figures. Here you look at theta frequency that increases with running speed in the real world. In VR, theta frequency actually drops very quickly from stationary to low speed and then it remains flat. The amplitude of eta in the VR shows a nonlinear dependence. It drops first and then goes up, whereas theta amplitude in the eta band signal, of course, keeps reducing. Now, who is doing it, which is sitting around in the cell layer? Those are the inhibitory interneurons. If we looked at the autocorrelation, which counts how rhythmic the neural activity is in the real world, these are 30 different CA1 interneurons in the real world. They show maybe 2 peaks, and after that, there is nothing, suggesting the rhythmicity kind of decays after 200 to 300 milliseconds. Here are several interneurons, and as you can see, the rhythmicity keeps going. This is 1, 2, 3, 4, and it still keeps going on. You can look at the autocorrelations averaged across many, many cells. All of these, you see very strong increase in theta rhythmicity. That's shown here in different ways. And the reason this happens is that the preferred phase of the interneuron spiking with respect to theta and eta is highly correlated. And so is the depth of modulation of a cell in virtual reality. This is not seen in the real world. So it is these interneurons which contain GABA, which exert huge force on CA1 pyramidal neurons. They are the ones which seem to be driving this novel rhythm and dynamics in the CA1 cell layer. So we conclude from this that there are several neurocognitive disorders that are related to loss of theta or change in theta frequency. We have shown in previous theoretical papers that actually when you induce neuroplasticity, if the signal is very periodic, a specific frequency can induce a lot of plasticity. Other signal of a different frequency induces less plasticity. And if you take away the rhythmicity, everything decreases quite a bit. So this shows that exact rhythmicity, not just the frequency, but the rhythmicity of a signal is quite important. And the precise frequency is important, and the results depend quite a bit on where the synapses are on the dendrite. We have shown that VR can reliably boost the 8 Hz theta rhythmicity and induce a novel 4 Hz theta rhythm. This raises the possibility that VR can be potentially used as a therapy to control or boost brain rhythms, neuroplasticity, and neural wiring, which can be useful for treating many learning and memory disorders.